So today we're going to talk about carbohydrates, and by the end of this lecture you should be able to say that you can use basic nomenclature to describe glycans of various types, recognize Fisher and Haworth projections and match them to the 3D structure of the cyclic monosaccharide, know the difference between alpha and beta linkages, predict the products of several reactions which include mutarotation, glycosylation, and glycosylation reactions, reduction reactions of glycans, oxidation of glycans to the corresponding carboxylic acids, and oxidative cleavage of diols. Also be able to draw mechanisms for the above reactions. Carbohydrates are the most abundant organic compounds in the plant world. Glucose, starch, and glycogen act as storehouses of chemical energy. Cellulose forms the supportive structures of plants. Crustacean shells are made of chitin, and glucosaminoglycans form connective tissues in animals. Ribose and deoxyribose are essential components of nucleic acids, which are the building blocks of DNA. On this slide, you can see a few examples of complex carbohydrates ranging from sweeteners like sucrose, which is table sugar, sucralose, Splenda, and ribaudioside A, which is the natural product extract of stevia, the sweetener in stevia, all the way to digitoxin, which is a compound isolated from foxglove that has a steroid core and then a carbohydrate portion here. And this molecule is used in cardiology to reduce pulse rate, regularize heart rhythm, and strengthen heartbeat. And then all the way to the infamous spike glycoprotein of COVID-19, shown here on this virus picture, which you're probably all familiar with by now. This glycoprotein binds to the human cellular receptor ACE2, and it's responsible for the entry of the virus into the cell. So here you can actually see the spike glycoprotein. It's a trimeric structure, so each of the monomers are shown here in a different color. The main colored part is the protein part, and the little red parts that look kind of like a candy topping are the representations of the glycans on the surface. Monosaccharides have the general formula C2H2ON, with one of the carbons being the carbonyl group of either an aldehyde or a ketone. The most common monosaccharides have 3 to 8 carbon atoms. The suffix "-ose" indicates that the molecule is a carbohydrate, and then the prefix tells you how many carbons are in the structure. So a triose is made of 3 aldehyde units, such as glyceraldehyde here. Monosaccharides containing an aldehyde group are classified as aldoses, and those containing a ketone are called ketoses. So this is a keto triose, and it belongs to the bigger category ketose. There are only 2 possible trioses, and they are glyceraldehyde, the aldotriose, and dihydroxyacetone, a ketotriose. 1,3-dihydroxypropanone, more commonly known as dihydroxyacetone, is the active ingredient in artificial tanning agents such as Mantan and Magic Tan. Be careful when using these products. They smell weird, and you can definitely overdo it, as shown by this timeline of photos from 2004 to 2014. Monosaccharides are often drawn as the Fisher projection, and we use D and L descriptors for chirality rather than RNS. So here below I'm showing an example with R-glyceraldehyde. Glyceraldehyde contains a chiral center and therefore exists as a pair of enantiomers. The horizontal segments of a Fisher projection represent bonds directed toward you, while the vertical segments represent bonds that are directed away from you. Only the atom in the center, the chiral carbon, is in the plane of the paper. The configuration of carbohydrates, as well as those of amino acids and many other compounds in biochemistry, is commonly designated by the DL system proposed by Emil Fischer in 1891. At that time, it was known that one enantiomer of glyceraldehyde had a specific rotation of plus 13.5, and the other had a specific rotation of minus 13.5. Fisher proposed that these enantiomers be designated D and L for dextro-rotary and levo-rotary, respectively, but he had no experimental way to determine which enantiomer had which specific rotation. Fisher therefore did the only possible thing. He made an arbitrary assignment. He assigned the dextrorotary enantiomer an arbitrary configuration and named it D-glyceraldehyde. He named its enantiomer L-glyceraldehyde. These assignments were eventually established to be correct using a specialized X-ray crystallographic technique. D and L-glyceraldehydes serve as reference points for the assignment of relative configuration to all other aldoses and ketoses. The reference point is the chiral center that's farthest from the carbonyl group. Because this chiral center is always the next to last carbon in the chain, it is called the penultimate carbon. Note that for monosaccharides with 2 or more chiral centers, the designations D or L refer only to the configuration of the highest numbered chiral center. Also note that the D or L designation of a given monosaccharide does not specify the sign of the specific rotation of the compound. If the sign of the rotation of plane polarized light is to be specified in a name, it is given by a plus or a minus sign preceding the name. This is the same as in R and S in traditional small molecules. This chart shows names and Fisher projections for all tetroses, pentoses, and hexoses. Each name consists of 3 parts. The letter D specifies the configuration of the penultimate carbon. Prefixes such as rib, arabin, and gluc specify the configuration of all other chiral centers in the monosaccharide. The suffix -ose shows that the compound is a carbohydrate. Amino sugars contain an amino group in place of a hydroxy group. Only 3 amino sugars are common in nature: glucosamine, minosamine, and galactosamine. N-acetylglucosamine, also called glicnac, is a derivative of glucosamine and is a component of many polysaccharides, including chitin, the hard shell-like exoskeleton of lobsters, crabs, shrimp, and other shellfish. It is also involved in modulating a variety of biological pathways with impact in diseases like cancer, diabetes, and neurodegeneration, and is also involved in epigenetics. Many other amino sugars are components of naturally occurring antibiotics. Here we have selected aminoglycoside antibiotics, which are highly important treatments against gram-negative bacterial infections. Due to widespread clinical use, there is significant resistance to the parent compounds paromomycin and neomycin. Additionally, they cause nephrotoxicity or liver damage, which can be managed by dosing parameters, as well as drug-induced ototoxicity or hearing loss, which affects between 20 and 37% of patients due to damage to hair cells in the cochlea. Severe hearing loss is experienced in patients with a mutation at adenine 155, where it's converted to a guanine. Through collaborative studies with several labs, including Christine Chow's lab here at WSU, a former Wayne State faculty member, David Kreitch, and his co-workers have made significant progress in the rational design and chemical synthesis of analogs of apomycin, paromomycin, and neomycin. They've developed molecules that bind preferentially to the RNA of the targeted bacteria and minimally to the human mutant A1555G, which is predictive of an eventual drug that would avoid the problematic hearing loss side effect. More recently, they've developed an analog called propylomycin that has an enhanced interaction with adenine 1408, which increases its potency against antibacterial strains. The replacement of a hydroxy group on the sugar with a propyl group enhances the antibacterial activity, as well as protecting against the AMEs, or aminoglycoside modifying enzymes, that act on ring 1. This modification should circumvent several common mechanisms of aminoglycoside antibiotic resistance And it also is responsible for the increased ribosomal selectivity and the resulting reduction in autotoxicity. Monosaccharides are colorless crystalline solids, although they often do crystallize with some difficulty. Because hydrogen bonding is possible between their polar hydroxyl groups and water, all monosaccharides are very soluble in water. They are only slightly soluble in ethanol and are insoluble in nonpolar solvents such as diethyl ether, chloroform, and benzene. We saw that cyclic hemiacetals form very readily when hydroxyl and carbonyl groups are part of the same molecule, and their interaction can form a 5 or 6 membered ring. For example, 4-hydroxypentanol forms a 5-membered cyclic hemiacetal. Monosaccharides have hydroxyl and carbonyl groups in the same molecule. As a result, they too exist almost exclusively as 5-membered and 6-membered cyclic hemiacetals. A common way of representing the cyclic structure of monosaccharides is the Haworth projection, named after the English chemist Sir Walter N. Haworth, who won the 1937 Nobel Prize in Chemistry. In a Haworth projection, a 5 or 6-membered cyclic hemiacetal is represented as a planar structure lying perpendicular to the plane of the paper. Groups bonded to the carbon of the ring then lie either above or below the plane of the ring. The new chiral center that's formed during the cyclization is called the anomeric carbon. Stereoisomers that differ in configuration only at the anomeric carbon are called anomers. These are just a special type of diastereomer. The anomeric carbon of an aldose is carbon 1. That of fructose, the most common ketose, is carbon 2. Haworth projections are most commonly written with the anomeric carbon to the right and the hemiacetal oxygen to the back. In the alpha form, the exocyclic oxygen group at the anomeric center is on the opposite face to the CH2OH group. And in the beta form, the exocyclic oxygen group at the anomeric center is on the same face as the CH2OH group. If a mixture of the alpha and beta anomers are present, then this is often represented using a wavy line to represent the bond. Here I've drawn it out with glucose, where we have a wavy line showing that we have a mixture of the alpha and beta isomers. Pyranoses are best represented as their chair forms, as this better conveys stereochemical and reactivity information. In larger carbohydrates, this view also conveys the overall molecular shape, as you can see for amylose here. An important process to understand from a mechanistic perspective is mutarotation. This is the process by which monosaccharides interconvert in between anomers. Pause the video and then, using your knowledge from Chapter 16, propose a mechanism for this interconversion, first using a catalytic base, then using catalytic acid. A glycosylation reaction is essentially just an acid-catalyzed acetal formation where you start at the hemiacetal stage of the mechanism. I'll draw this mechanism out here. First, we'll protonate the anomeric hydroxyl group. Then we'll kick out water, and we'll use this lone pair to assist in that process to form an oxocarbenium ion. Water can then add back in, but from the opposite face of the glycan. Sorry, that's methanol in this reaction. So methanol will add in from the opposite face of where the activated anomeric hydroxyl departed. And that'll lead to an intermediate shown here. A final deprotonation step will lead to the glycosidic bond of the alpha anomer there. If the methanol in this step adds to the top face where the water had just departed, that would then lead to the beta anomer. In a simple pyranoside, The beta anomer will be equatorial relative to the ring, and then the alpha anomer will be axial. So that's a handy mnemonic for alpha is axial. Since we also know that this alpha group is on the opposite face as the CH2OH, that can help us to remember the more general rule about alpha and beta anomers, that the alpha anomer has the groups oriented on opposite sides of the plane of the ring. Just a final note that glycosidation is actually different than glycosylation, which is the process of attaching a glycan to peptides, proteins, or lipids. Because pyranoses are in equilibrium with aldehydes, they can be reduced with agents that reduce aldehydes as well. Here we have sodium borohydride, which is a good choice for a glycan because sodium borohydride reductions are normally performed in alcoholic solvents. So all these alcohol functional groups will be tolerated by the borohydride reagent. This mechanism is as you learned it in chapter 16. So you can pause the video and draw it out here. In a basic aqueous solution, aldoses can be oxidized to aldonic acids or uronic acids. The aldonic acid, which is what we're seeing on this slide, will involve first hydration of the aldehyde, followed by oxidation of that hydrate to form the carboxylic acid. Due to the basic reaction conditions, this carboxylic acid will be rapidly deprotonated to form the carboxylate. Then an acid workup would be required to isolate this carboxylic acid. So pause the video for a second and think about what oxidations you know that could perform this reaction. Oxidation to form a uronic acid requires an enzyme to complete. Enzymes are special because by appropriate positioning of molecules in their active sites, they can perform reactions that are difficult to impossible in a reaction flask. Here, an enzyme converts a primary alcohol at C6 of the sugar to a carboxylic acid. This carboxylic acid is known as a uronic acid. Because the aldehyde is not in the active site of the enzyme with the oxidizing group, the aldehyde will not be oxidized to the carboxylic acid even if a hydrate is formed. The final reaction to discuss from this chapter is the oxidative cleavage of diols by periodic acid. We'll follow the simple steps of first, attach the alcohol or alcohols to the oxidant, and second, shuttle electrons in a cyclic transition state to try to give electrons to the oxidant. So we'll start by attaching the diol to the oxidant. We have the alcohol add to the iodine, and we can push one of these pairs of electrons up onto the carbonyl. So that'll lead to this intermediate, and then we'll get a proton transfer to give this product. Reforming the iodine-oxygen double bond allows us to kick out water. And now addition of the second oxygen leads to the next intermediate. So then a proton transfer is going to get us to a neutral intermediate, and that's where we're going to have our electrons moving in a circle to give electrons to iodine. So this is the key thing when you're doing an oxidation, is to think about how to give electrons to your oxidant because it wants to be reduced. Okay, so if we draw that arrow, that means that we need to draw an arrow that will replace electrons on this oxygen from which we stole electrons. So we're going to do that by breaking the carbon-carbon bond. If we do that, we're going to have a plus at the other carbon here, and so we prevent that by breaking the iodine-oxygen bond. So then we end up with two aldehyde groups and iodic acid as our byproduct. So with the number of hydroxyl groups that are side by side, or vicinal in a sugar, you can imagine that quite a lot of destruction happens when you do the proiodic acid cleavage of a hexose, for example. So I hope you enjoyed that introduction to carbohydrate chemistry. We'll finish up next time with a video talking about different sizes of carbohydrates, ranging from disaccharides to oligosaccharides to polysaccharides.